We live in an age of incredible scientific discovery. Every day, researchers are pushing the boundaries of what we know and what we can achieve. The pace of innovation is accelerating, and the implications for our future are profound. The human genome has been mapped, a monumental achievement that has opened up new avenues of research and understanding. This map is like a blueprint, detailing the intricate design of human life. We are unlocking the secrets of DNA, the very code that defines who we are. Each discovery brings us closer to understanding the complexities of life at a molecular level. And now, with CRISPR-Cas9, we have a tool that allows us to edit that code, the very blueprint of life itself. This technology is revolutionary. It gives us the power to make precise changes to DNA, potentially correcting genetic defects and enhancing biological functions. It holds the potential to cure diseases, enhance crops, and reshape the world around us. Imagine crops that can withstand harsh climates or medical treatments tailored to an individual's genetic makeup. But with such power comes profound responsibility. The ability to alter the genetic code of living organisms raises significant ethical questions. We must consider the long-term consequences of our actions. Imagine a world without genetic diseases. Families could be free from the fear of passing on debilitating conditions to their children. The quality of life for millions could be dramatically improved. CRISPR makes this a real possibility. By targeting specific genes, we can potentially eliminate conditions that have plagued humanity for generations. We could potentially eliminate cystic fibrosis, Huntington's disease, and even some forms of cancer. The medical applications are vast and could revolutionize how we approach treatment and prevention, and even some forms of cancer. The potential to save lives and reduce suffering is immense. The implications for human health are staggering. We are on the brink of a new era in medicine, where genetic information can guide personalized treatments and interventions. But the ability to alter our genes raises ethical questions that we must confront. What are the moral implications of editing human DNA? How do we ensure that this technology is used responsibly and equitably? What are the limits of this technology? Should there be boundaries on what we can and cannot change? These are questions that society must grapple with as we move forward. And who gets to decide how it's used? Policymakers, scientists, and the public must come together to create guidelines and regulations that balance innovation with ethical considerations. The power to edit genes is not just limited to humans. CRISPR can be used to engineer plants and animals, potentially transforming agriculture and food production. CRISPR can be used to engineer crops that are more resistant to disease and drought. This could lead to higher yields and more sustainable farming practices. This could help us address global hunger and food security. By creating crops that can thrive in challenging environments, we can ensure a more reliable food supply for the world's growing population. But introducing genetically modified organisms into the environment also carries risks. We must carefully assess the potential impact on ecosystems and biodiversity. We need to carefully consider the potential impact on biodiversity and ecosystems before we unleash this technology on a large scale. The balance of nature is delicate, and unintended consequences could be far-reaching. The CRISPR revolution is here. It is a powerful tool that holds great promise, but also demands careful consideration and responsible use. It is time for a global conversation. We must come together as a global community to discuss the implications of this technology and how it should be governed. We must engage with the ethical, societal, and environmental implications of this powerful new tool. Open dialogue and collaboration are essential to navigate the challenges and opportunities that lie ahead. The future of life on Earth may depend on it. The choices we make today will shape the world for generations to come. Let us approach this new frontier with wisdom, caution, and a commitment to the greater good. CRISPR-Cas9 works like a pair of molecular scissors, a revolutionary tool in the field of genetic engineering. These molecular scissors are guided by a piece of RNA that matches a specific DNA sequence, ensuring precision in the editing process. This allows scientists to target and cut out specific genes with remarkable accuracy, paving the way for targeted genetic modifications. Once the gene is cut, the cell's natural repair mechanisms kick in, attempting to fix the break in the DNA strand. Scientists can then introduce new genetic material at the site of the cut, effectively editing the genetic code. This technology is groundbreaking and holds immense potential. CRISPR is faster, cheaper, and more precise than previous gene editing techniques, making it a game changer in the field of genetics. It has the potential to revolutionize medicine by providing cures for genetic diseases, enhance agricultural practices by creating more resilient crops, and even deepen our understanding of the fundamental building blocks of life itself. But the ease with which we can now alter the building blocks of life raises serious ethical concerns that cannot be ignored. One of the most controversial applications of CRISPR is germline editing, which involves making changes to the DNA of embryos, sperm, or eggs. This process involves altering the genetic material in a way that these changes would be inherited by future generations. These genetic modifications would then be passed down to future generations, potentially altering the human gene pool permanently. Germline editing could potentially eliminate genetic diseases from a family line forever, offering hope to those affected by hereditary conditions. However, it also opens the door to the possibility of designer babies, where parents could choose their children's traits, such as intelligence, physical appearance, and even personality. The ethical implications of germline editing are profound and complex, raising questions about the morality of such interventions. Do we have the right to alter the genetic makeup of future generations? What are the moral boundaries of such power? What are the potential unintended consequences of such modifications? Could we inadvertently introduce new genetic problems? These are questions that we must grapple with as a society, engaging in thoughtful and informed debates about the future of genetic engineering. The decisions we make today regarding the use of CRISPR technology will have far-reaching implications for the future of humanity, shaping the genetic destiny of our species for generations to come. The idea of designer babies has long been a staple of science fiction, but with the advent of CRISPR, it is moving out of the realm of fantasy and into the realm of possibility. Imagine a world where parents could select their child's eye color, height, or even intelligence. This may sound appealing to some, but it raises a host of ethical concerns. One concern is that germline editing could exacerbate existing social inequalities. If wealthy parents could afford to enhance their children's abilities, it could create a genetic underclass. This would further entrench existing power structures and undermine social mobility. Access to this technology must be equitable if we are to avoid creating a genetic divide. Another concern is the potential for unintended consequences. Our understanding of the human genome is still incomplete. Making changes to the germline could have unforeseen and potentially harmful effects on future generations. We must proceed with extreme caution to avoid unintended consequences that could harm future generations. The potential for germline editing to eliminate genetic diseases is undeniably appealing. However, the ethical implications of this technology are profound. We must carefully weigh the potential benefits against the risks before we embark on the path of human germline editing. The future of our species hangs in the balance. The human genome is a complex and interconnected system. It is composed of over 3 billion base pairs of DNA, which encode the instructions for building and maintaining a human being. Each gene within this vast network plays a specific role, and the interactions between these genes are intricate and finely tuned. Altering one gene can have ripple effects throughout the genome, leading to unintended consequences. These changes can cascade through the genetic network, affecting multiple pathways and processes in ways that are difficult to predict. These consequences could be benign, such as minor changes in physical traits, but they could also be harmful or even fatal. For instance, a seemingly small mutation could disrupt a critical biological function, leading to severe health issues. We simply don't have enough knowledge about the intricacies of the genome to predict all the potential outcomes of gene editing. Our understanding of genetic interactions is still in its infancy, and the full scope of genetic regulation and expression is far from being completely mapped out. One concern is that gene editing could lead to the creation of new diseases or the resurgence of old ones. By altering the genetic makeup of an organism, we might inadvertently create vulnerabilities that pathogens can exploit. For example, a gene edit intended to confer resistance to one virus could inadvertently make a person more susceptible to another. This could happen if the edited gene plays a role in the immune response to multiple pathogens. Or, a gene edit designed to eliminate a particular genetic disorder could have unforeseen negative consequences on other aspects of health. The interconnected nature of the genome means that changes in one area can have far-reaching effects. Another concern is the potential for off-target edits. These are unintended changes to the DNA that occur when the gene editing tool, such as CRISPR, makes cuts at unintended locations. CRISPR is a powerful tool, but it is not perfect. While it has revolutionized the field of genetics, its precision is not absolute, and off-target effects can occur. Sometimes the Cas9 enzyme can cut DNA at unintended locations in the genome. These off-target cuts can disrupt the function of important genes, leading to unpredictable health problems. These off-target edits could disrupt the function of important genes, leading to unpredictable health problems. This could result in new genetic disorders or exacerbate existing conditions. Scientists are working on ways to improve the precision of CRISPR, but off-target edits remain a concern. Advances in technology and a deeper understanding of the genome are needed to minimize these risks. The potential benefits of gene editing are vast. It holds the promise of curing genetic disorders, eradicating diseases, and even enhancing human capabilities. But we must proceed with caution and humility. The power to alter the human genome comes with great responsibility, and we must ensure that we fully understand the implications of our actions. We are still unraveling the mysteries of the human genome. Each discovery leads to new questions, and the complexity of genetic interactions continues to challenge our understanding. Until we have a more complete understanding of the potential risks, we must exercise restraint in our application of this powerful technology. Ethical considerations and rigorous scientific research must guide our efforts. The health and well-being of future generations depend on it. By carefully considering the consequences of gene editing, we can ensure that we use this technology to benefit humanity without causing unintended harm.